Okay, thank you very much for coming. So I'll continue today about this relatively new approach that our lab has taken, and this is as I talked in the previous, in my third lecture, a genetic and proteomic approach to understand the function of cell cycle proteins. In my previous three lectures, I introduced to you the cell cycle machinery and the proteins that drive cell cycle progression. We talked about the D-type cyclins on the first lecture and about E and A-type cyclins in the second one. And then I talked about the function of cyclin in the brain in quiescent neurons in the third lecture. In my last lecture, I focused on cyclin D1. Once again, this is one of the earliest cyclins that is being induced. It binds and activates cyclin-dependent kinase 4 and phosphorylates the retinoblastoma protein, and this leads to release of V12 transcription factors, which are required for S-phase entry. Cyclin E is also one of the targets of V12s, and this is kind of a feedback loop that triggers entry of the cells into the S phase. Cyclin D1 is, I'm also repeating what you already heard, is that among all the human cancer genes, cyclin D1 is the second most frequently amplified gene in human cancer genome. And for this reason, we are very interested in this protein, and we want to understand the full repertoire of the functions of these proteins in the living organism in cancer. Clearly, the function that I just described to you, the ability to bind cyclin-dependent kinase 4 and phosphorylate the retinoblastoma protein is a very important one. However, we want to understand the full repertoire of those proteins. And I did mention in my third talk is that the technique that we use routinely is we take our protein, we express it in a cell, and then we attach two short sequences that encode the tags called the flag tag and hematoglutinol HA tag, and then we make the lysate of these cells. We put it through the column containing anti-flag antibody. Theoretically speaking, all the protein should flow through. Only your protein should be kept because we attach flag tag. In practical terms, however, there is lots of contaminants. They stick. We elute this complex with the flag peptide, and then we load into the second column, which contains anti-HA antibody. Since we attach also HA tag shown here in red, our protein together with complexes is going to be kept. We can just elute it, run in silver stain gel, and either cut out the individual bonds in a kind of more old-fashioned way, or do mass sequence everything in a shotgun mass spec sequencing. However, Fred Benveni, a postdoc in the lab, wanted to understand the function of cyclin D1 in vivo. This is the section of the embryo where we are detecting cyclin D1 messenger RNA, and this is in red. As you can see, the highest level of cyclin D1 is in the retina. In fact, the level of cyclin D1 in the retina is so high that all other tissues seem to be negative. They are not negative. This is just, you know, this exposure time. When you have a clear signal here, this is the same like with Western blotting. If you have a very strong band, you expose it for a short time, you have a very strong band in lane 1, and lane 2 and lane 3 looks like negative, right? If you expose it longer, you start seeing bands. So cyclin D1 is expressed in many compartments, but in the retina is particularly high. It's also very high in the brain, and also postnatally when females become pregnant, it's induced in mammary glands, and lastly when there's a cancer, breast cancer arising in mammary gland, there is once again a high level of cyclin D1. So the Fred's project was he wanted to know what cyclin D1 partners right here, and what are the partners right here, and what are the partners right here. And I should add that the retina is a very important one, because not only cyclin D1 is highly expressed there, but is required for retinal development. Because we talked here a little bit, well, why those cyclins were developed in evolution if we really don't need them. Well, actually, retina is a good example. There is very high level of cyclin D1, and the retina depends on cyclin D1. If you knock out cyclin D1, the retina becomes hypoplastic. So to address this question, Fred generated a knock-in strain of mice where, using gene targeting, he inserted into the 5' end of cyclin D1 gene the sequences encoding flag and HA tags. So this is one strain of mice which express in a homozygous state cyclin D1, which in the N-terminus, amino terminus, contains flag and HA tags. Now, of course, you realize, and also Fred realized, that if you put target one end of the molecule, that this might actually obscure some interactions. Because if the interaction occurs through the N-terminus, then by putting those tags, you might lose some interactions. And Fred, of course, wanted to capture all interactions. So, and therefore, he generated the second strain of mice, where he put those tags at the C terminus. So it's a completely different strain of mice, one containing tag cyclin D1 and N terminus, and the second in a C terminus. And then he wanted to characterize whether everything is okay with the tag cyclin D1. So, for example, here you can see a Western blot for cyclin D1 in control mice, and in heterozygotes we have two bands. This is endogenous cyclin D1, this is tagged cyclin D1, which is migrate slightly higher because we attach those tags. This is the C tag, this is the N-terminal tag. If you probe it with the antibody against the tag, you only detect it in tagged mice, but not in untagged mice. And if you immunoprecipitate cyclin D1, you can have the kinase activity both from wild type, from cyclin D1, C tag and N tag. So they are pretty much, there's pretty much wild type biochemically. I also told you, so this is the wild type retina. I also told you that if you knock out cyclin D1, the retina is hypoplastic, it doesn't develop normal. But if we express cyclin C that has a C tag, then now this retina looks normal. And if you have the cyclin D1 that is N tagged, also the retina is normal. So this tagging of cyclin D1 does not do anything. It maintains the wild type protein, because this protein can drive normal development of the retinal. I also mentioned to you that, so this is the wild type mammary gland, this is the mammary gland of cyclin D1 knockout, is hypoplastic, but if you have cyclin D1 that is C tagged or N tagged, the mammary gland is normal. So putting those tags did not do anything to the protein. This is a really wild type protein that not only behaves wild type biochemically, as shown here, for example, by comparing the kinase assay of wild type versus N tag or C tag, but also is wild type developmentally by being able to drive normal retinal and normal breast development. And because Fred wanted to have the full repertoire of the interactions, and as I mentioned to you, the N tag or the C terminal tag might disrupt some interactions, what he did is that he took homozygous N-tagged mice and homozygous C-tagged knockout mice and crossed them. So the next generation here is what he calls hybrid mice, when in every cell one copy of cyclin D1 is N-tagged and the second is C-tagged. So 50% of the molecules are going to contain tag on this end and 50 on this end. And this is a trick, so we can capture all the interactors. If we just had everything on one end, then theoretically we might lose some interactions. But because we have a hybrid mice where one copy, 50% of the protein is tagged here and 50 is tagged here, then even if we are losing some interactions here, we are going to capture them here and vice versa. And then he took different organs and not the cells, but he took actually the retinas. I think it's shown here. So he prepared the retinas, the brain, the mammary gland, and breast cancer, and then he purified cyclin D associated proteins. And you can see here that this arrow, the star corresponds to cyclin D1, and each of these proteins is a different cyclin D1 interacting proteins. And as you can see, cyclin D1 interacts with different proteins in the retinas, that's for example in mammary gland or in breast cancer. And then we did mass spec sequencing, and then for the purpose of today's talk, I'm not going to give you the list of all 4 lists, but I'll just say that all those interactors we put into gene ontology analysis, which tells us which functions are enriched there. And of course we're expecting to see proteins in a cell cycle category, because cyclin D1 is a cell cycle protein, and sure enough this was the most enriched function among cyclin D1 interactors. But quite surprisingly, we found that the second most enriched group is transcription. And then we found several transcriptional proteins interacting with cyclin D1. This was not totally surprising, because there have been reports in literature that cyclin D1 can bind transcription factors. And actually it was proposed that cyclin D1 can serve as a bridge between transcription factor and chromatin modifying enzymes. So we wanted to, Fred said, okay, well, if cyclin D1 binds transcription factors, then it's possible that cyclin D1 is actually sitting on a DNA, like transcription factors. And if you want to interrogate the interaction of your protein with the genome, what you do is that you do something that at that time it was called chip, although now there's a more modern version called chip-seq. So I'll tell you this more old-fashioned way that we used, and then I'll tell you about the more modern way. So the old-fashioned way is chip is done as follows. You want to see where your transcription factor is sitting in the genome. And therefore what you do is that you cross-link your all the proteins to DNA, and of course, among other things, you are going to cross-link your transcription factor to DNA. And then you shear the DNA into pieces. So only a small piece of DNA is going to be bound to your transcription factor, and then if you have a good antibody against your transcription factor, you can immunoprecipitate. And now, in the old-fashioned way, you can hybridize it to the array, which contains the whole genome. So you can know exactly where your transcription factor is binding. In the more modern way called ChIP-seq, you just sequence this DNA. And this way you know where your transcription factor is binding. Now in our case, and of course you need to have some control. Because if you immunoprecipitate your transcription factor, of course you have the specific sequencing, but you have a junk. And usually as a control you use some non-specific immunoglobulin. In our case, it was a little bit more complicated. We didn't want to do it from the in vitro cultured cells. We wanted to do it right here from the retina. And we expected that cyclin D1 is not sitting in the DNA, but is actually sitting on transcription factors which sit on DNA. And therefore, Fred, number one, had to scale down this procedure because we, of course, the embryonic retina is very small. And number two, he had to change this cross-linking procedure so he can cross-link cyclin D1 to transcription factor to DNA. So when he is immunoprecipitating cyclin D1, he's going to then immunoprecipitate transcription factor and then immunoprecipitating the DNA. On the other hand, we had a very good advantage because our cyclin D1 was tagged, right? I just showed you those slides that we put flag and HA tags. And actually, these are very good antibodies for the ChIP and ChIP-seq. So what we could do is we could take our knock-in embryo, and from this knock-in embryo, we would immunoprecipitate cyclin D1 together with the DNA using anti-flag antibody. Because our cyclin D1 is tagged, then we could use the very same antibody, anti-flag antibody, from wild-type embryo. Because wild-type embryo, everything is the same, all the proteins is the same, the DNA is the same, but there is no flag-tag cyclin D1, right? So we can so immunoprecipitate cyclin D1 with anti-flag antibody from our knock-in embryos, and we label it with one color. We label it with red. And then we take we IP cyclin D1 from wild-type mice, which are exactly the same, but since we are using anti-flag antibody, there is no flag-tag cyclin D1 in wild-type mice, so this is the background. We label it in green, and then we mix it and hybridize onto the array. So whatever is going to be enriched into the first IP is specific, right? Whatever is going to be red, whatever is going to be enriched in anti-flag immunoprecipitation from the knock-in embryo is going to be specific. And you can do exactly the same thing with anti-HA antibody, which once again you immunoprecipitate cyclin D1 from anti-HA antibody from knock-in embryo, so that is a specific signal, one color. You immunoprecipitate with anti-HA antibody from wild-type embryo, this is the background, and then you hybridize it together. And when we did it, we immediately noticed that there was so this is intensity in a wild type in a wild type embryo, and this is the knock-in embryo, and everything here has the same intensity. And this is this bump. These are the sites. These are the promoters that are specifically bound by cyclin D1 which are enriched in anti-flag IP from the knock-in embryos. So we could then go to these genes, we could go to the promoters that are bound by cyclin D1, and we could ask some global bioinformatics questions about what's special about the promoters of the genes that cyclin D1 is sitting in the retina. So the first question we asked is that we asked what's the fraction of the CpG, what's the frequency of CpG dinucleotides in our promoters? And the reason being that the promoters that are rich in the CpG islands are generally highly expressed. The ones that have a low CpG content, they are poorly expressed. So as you can see, this is the distribution of CpG content in the promoters that are bound by cyclin D1, most of them being somewhere here. And this is the distribution of promoters not bound to cyclin D1. So you can see this distribution is skewed to the right. There is more, there is a higher there is a higher CpG content in the promoters that are bound by cyclin D1 rather than promoters that are not bound by cyclin D1. So cyclin D1 is binding promoters that are high in CpG content, which suggests that this could be binding a highly expressed genes. Actually, in the retina, there is a very good way to judge the expression of genes, because they are the so-called stage libraries. And stage libraries is basically the corresponds now more modern version is RNA sequencing, where for every transcript you know what's the abundance of this transcript. You can check how many tags there are in the library. The more tags, the more abundant is the transcript. So in this experiment, which doesn't project well because of the color, but I hope you will get, here we are taking some of the, so each line corresponds to a different gene, and this dark color indicates binding. So this is transcription start site. There is no binding, there is no binding, 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 no binding. And these are, so these are the top 100 genes that are bound by cyclin D1. And these are 100 genes that are not bound by cyclin D1. And then for every gene, we are showing you not only the binding, but when you go to this column, this is the stage count, meaning how many tags are in the library. Many tags meaning highly expressed, little tags meaning not highly expressed. And the highly expressed genes are in yellow, and the poorly expressed genes are in blue. And as you can see, there is more yellow here than here. There is more blue here. So of course we did some statistical analysis, but even visually, and I'm sorry, the color does not project well, but I think even in this poor projection you can see that if we look at the genes that are bound by cyclin D1, they are more likely to have a yellow number of tags, meaning be highly expressed. If you look at the genes that are definitely not bound by cyclin D1, they are more likely to be blue genes, and therefore they're not well expressed. So for this, together with CpG content, suggests that cyclin D1 is sitting on the promoters of actively expressed genes. Of course, this is only a trend. You can, among cyclin D1 bound genes, you can have some blue lines, and among the cyclin D1 not bound genes, you can see yellow lines. So of course, cyclin D1 is not a marker of active genes, but is enriched on genes that are actively transcribed. And then this is you can actually of course with this procedure you see where the binding is, because this is the hybridization across the promoter. This is the transcription factors of the gene, and you can see here a very nice peak binding of cyclin D1. This is another gene, and it's right on the transcription start site. This is the third gene, and this is the fourth gene. So those, just for examples, indicate that, at least in those 4 cases, the binding is very close to the transcription start site. These are, of course, 4 examples, but I'll now show you the data of the all binding events of cyclin D1. When this is the transcription start site, this is upstream, this is upstream, and this is the density or the fraction of the cyclin D1 bound site. So as you can see, the overwhelming majority of cyclin D1 binding sites are around the transcription start site. So now with this knowledge, what we could do with the help of our bioinformatics colleagues, we could take all the sequences that cyclin D1 is binding, all those sequences, and we can ask, can we recover any particular sequence? So one possibility to cyclin D1 is recognizing some specific DNA sequence. In this case, what we were hoping is when we put all those sequences into the database to come up with some consensus sequence. Alternatively, as proposed in the literature, cyclin D1 might not bind DNA directly, but through the transcription factors. So in this case, we would not get just one sequence, but many sequences of several different transcription factors. And the latter was the case. We didn't get one sequence, we got several enriched sequences, which are actually shown here. These are enriched sequences for cyclin D1 binding. This is just a few of them, but each of them corresponds to different transcription factor that is binding DNA. And actually, by immunoprecipitation Western blotting, we verify that in retinas, cyclin D1 can bind to each of those transcription factors. So these transcription factors are binding DNA, and cyclin D1 is sitting on them, and is doing something. So now we have to find out what cyclin D1 is doing. And you know, the fact that what became obvious from analysis of using this chip as we use for chip-seq is that transcription factors are sitting on several target genes, but not every target gene that is occupied by your transcription factor is actually controlled by this transcription factor. The transcription factor plays a rate-limiting role in these genes. We wanted to know which genes are bound by cyclin D1 in a retina, so that we know, right? We have this long list, but we wanted to know which of those genes change expression if cyclin D1 is lost, because these are the ones that not only cyclin D1 is sitting, but it's actually doing something, right? So you have one list of genes where cyclin D1 is sitting. We know that this is direct interaction of cyclin D1 with the genome, but we don't know whether cyclin D1 is rate limiting or not. We have the list of genes which change expression where cyclin D1 is lost, but it could be direct transcriptional function or could be something direct. We want to intersect the two. We want to put together these genes, the genes that cyclin D1 is sitting on the promoters, and the gene that change expression. So we make gene expression profiles. Each lane corresponds to, I think, 10 different animals: wild type, wild type, wild type, D1, D1, D1 knockout. So you can see that each lane corresponds to the transcript. This transcript is high in wild type and low in cyclin D1 knockout. This is low in wild type, high in cyclin D1 knockout. If cyclin D1 is sitting on the promoter of this gene and the promoter changes the activity when cyclin D1 is knocked out, well, then there is a reasonably good chance that cyclin D1 is doing something. And this way we could narrow down the list of cyclin D1 bound promoters and control promoters to a short list. And why do we care? Why do we want to have this short list? Well, the reason being that, as I showed you, this is the retina of a wild-type mouse, and this is the retina of a cyclin D1 knockout mouse, and this is very hypoplastic. And we wanted to see that does this transcriptional function underlie this phenotype? Can this phenotype be due to cyclin D1 sitting on the promoters and doing something? So looking at the genes where cyclin D1 is sitting on a promoter, and which is ones which is turning, one of those genes is the notch gene. And notch gene is a very important gene which actually was shown to control to control the retinal development. Actually, in the notch-specific retinal specific knockout of notch, gives a very similar phenotype, suggesting that that there is at least some link between cyclin D1 and the notch expression. What we have found is that cyclin D1 is sitting on the notch promoter, and this is actually going from intersection. Not only cyclin D1 is sitting on the promoter, but actually notch expression goes down in cyclin D1 knockout, as if cyclin D1 was sitting on a promoter and upregulating notch expression. So first of all, we verified by the targeted chip that indeed cyclin D1 is sitting on the notch promoter. So this is notch promoter, and this is enrichment. This is actual PCR. And then just for myself, I would like to remind myself that notch is a transcription factor, is very unusual protein, which is normally in the membrane, and then it's cleaved by the gamma secretase, and goes to the nucleus, and it turns on the expression of target genes. In the retina, one of the targets, of the transcriptional targets of notch is Has5, and Has5 in turn represses those two basic key-lose proteins, MAT5 and NeuroD. So we have found that cyclin D1 is sitting on the notch promoter in the retina. We have found that we knock out cyclin D1, that there is less notch expression. So we wanted to see whether the whole pathway is affected. So if in cyclin D1 knockout retina, if you have notch less notch 1, you should have less Has5 and more of MAT5 in NeuroD, because less this meaning more this. This is repressing this. And then the case. This is the retina of cyclin D1 knockout mice. The Has5 is downregulated compared to wild type, and MAT5, the NeuroD, is upregulated. So this just shows that not only cyclin D1 is sitting on the notch promoter, not only if you don't have cyclin D1, you have less notch expression, but also you have less Has5, more MAT, and more NeuroD, suggesting that the whole pathways is under control of cyclin D1 in the retina. But this is all this is all correlation in the retinas. So now we are going to take the cell line, which is the retinal progenitor cell line, and then we are going to knock down cyclin D1, and now you can see that the level of notch goes down. So not only in the knockout retinas the notch is lower, but if you take the cell line and you knock down, you deplete cyclin D1, the notch goes down. And when you overexpress cyclin D1, the notch levels goes up. So this is consistent with the notion that cyclin D1 is controlling the level of notch. If you have less cyclin D1, you have less notch. If you have higher, if you overexpress cyclin D1, you have more notch. And in the retinas, we found that there is less transcription of the notch. But is there less protein? Of course, cells care about the protein. So this is a Western blotting, which each lane corresponds to I think 10 or 20 retinas. These are the wild type, but they are cyclin D1 knockouts. This is the loading control. This is cyclin D1. So this is the wild type retina has cyclin D1. The knockouts don't have cyclin D1, but importantly, the notch is greatly reduced in the retinas. So notch it's very well established that notch is a driver of retinal development. It's well established that if you knock out a notch in a retina, you have hyperplastic retinas, and we have shown that we knock out cyclin D1, we have less notch. So are you satisfied? Do you think that we have shown that cyclin D1 plays a transcriptional role by controlling the notch levels, and we can just write the paper and I can start the second lecture, or you are still not convinced? Ilya, are you convinced?
It's hard to say.
Hard to say, okay. Well,
occasionally good genes the same way.
Right. Now, I agree. This is correlation. This is consistent. It makes perfect sense. It could explain, but it's not the final proof. And how would you? How can you actually show that this is this phenotype is really responsible for the cyclin D1 knockout phenotype? That this is not, as you say, two independent things, but actually, yeah. Sorry.
Maybe express notch in cyclin D knockout.
Exactly. That's exactly what we did. Express notch in cyclin D1 knockout retina. So that's this experiment. It was done by actually an expert in the field, Koni Sepko, who is also in the Department of Genetics. And the way they do it is that this is a student, Karolina Mizeracka, and what they do is it's very kind of cool technique is that they can inject virus directly to the eye of a mouse, and this virus, the way it's injected, will go to the retinal cells, and then will infect the retinal cells. And the virus encodes beta-galactosidase, so this way later we can do dissection and stain for beta-galactosidase. So the blue cells are the ones that become infected, but still most of the cells will not be infected. And then we can see what happens with the proliferation of those cells. So cyclin D1 knockout the retinal progenitor cells proliferate very poorly. So if you just inject them, infect them with the virus, that then causes beta-galactosidase, so one cell becomes infected, and then you wait 10 days, and then you sacrifice animals, remove the eyes, and do the whole mount. Then you can see that 60% of the cells, of the blue cells, stay as a single cell, meaning they were injected, infected, and then the past 10 days they did not divide. About 30% of cells become two cells. So 30% of cells divided once in 10 days' time. And then 10% have actually 3 cells per clone, which is hard to rationalize because you could go from 2 to 4, right? Unless so this means they divided once, and then so you had 2 cells. And then one of those daughter cells divided, right? So now you can end up having 3 cells, and this is actually shown here. So these are the single cells. But if you put into the eyes virus encoding beta-galactosidase and notch, Well, now you restore proliferation. So you can see there are big clusters of cells. You know, even with the naked eyes, you can see that now there is a nice proliferation of the cells. And this quantification, there is very showing no cells did not divide, and most cells contain, most clusters contain over 4 cells, indicating that they started to divide. So I think that this provides some maybe convincing evidence that indeed the low level of notch is contributing to the phenotype, because if you raise the level of notch, that you can correct this proliferation deficiency. So we think that cyclin D1 is sitting on the promoter and is turning off the expression of notch. This is, let's imagine that this is notch gene. But cyclin D1, as I showed you, is not sitting directly on the DNA. It's sitting somewhere here on a transcription factor, and it was proposed to serve as a bridge between transcription factors and chromatin modifying enzymes. And actually in literature, that was proposed that cyclin D1 can bind P300 or CBP acetyltransferase. So using immunoprecipitation Western blotting, we ask in the retinas of mice, can cyclin D1 bind CBP? And the answer is yes. Here we can immunoprecipitate cyclin D1 and then Western blot for CBP, and we can see the complex. So so in the retinas, cyclin D1 is binding CBP. Of course, it doesn't mean that cyclin D1 and CBP is sitting on a promoter of the notch promoter. So now let's check this. So what we'll do, we'll immunoprecipitate cyclin D1 and PCR this promoter. And then if cyclin D1 is sitting there, then we should have enrichment. And that's the answer here. We immunoprecipitate cyclin D1, PCR the notch promoter, and we see that it's sitting there. Well, now let's ask whether CBP is sitting on a promoter. Now we are going to immunoprecipitate CBP, PCR the promoter, and see whether we see the complex. So we can do it. This is CBP, immunoprecipitate, and sitting on notch promoter. So can we conclude that CBP and cyclin D1 are sitting together on a promoter? We show that they bind by co-IP. We show that immunoprecipitate cyclin D1 is sitting on a promoter. If we immunoprecipitate CBP is sitting, can we say that they are sitting together?
It can be only one CD in each case.
Sure, it could be completely different pools. They can be some bound pool that is somewhere away, and they could be sitting on different promoters. So how we can check whether they are sitting together? Exactly. So that's the way to do it. You want to immunoprecipitate cyclin D, release the complex, immunoprecipitate CBP, and then PCR this promoter. And that's exactly done here. So immunoprecipitate cyclin D1, then we release the complex, and then re-IP with IgG, and there is nothing there, or IP with cyclin D1, re-IP with CBP, and then do the PCR, and they are sitting together. So IP cyclin D1, re-IP CBP, and PCR promoter, and we can see they're sitting together. So the expectation from if cyclin D1 is helping to bring CBP to the notch promoter, then we can make some expectations. First of all, if we knock out cyclin D1 or knock down cyclin D1, what do you think is going to happen with the recruitment of CBP? Do you think it's not going to change, go up or go down? What would be your expectation? Well, yes, if this is bringing CBP, it should go down. And then CBP acetylates those histones. So if we knock out cyclin D1, what do you expect to happen to acetylation of the histones around the notch promoter? Go up, go down, or not change? Go down. Okay, so let's see if you're right. So now we do in retinal progenitor cells. We knock down cyclin D1, or we overexpress cyclin D1, and we look at the recruitment of cyclin D1 to the promoter. So of course, there's no surprise. If you knock down cyclin D1, you have less cyclin D1 than the promoter, and if you overexpress, you have more. This is kind of a control. But here, if you knock down cyclin D1, you have less CBP recruitment, as you predicted. If you overexpress cyclin D1, you have more recruitment. This is the acetylation of the histone at the sites that are dependent on CBP. If you knock down cyclin D1, you have less. If you overexpress, you have more. This is less, this is more. This is in the retinal progenitor cells. This is knockdown experiment. I think it's nice because you can manipulate cells, but this is the cell line and with siRNA. Here is genetic experiment. I think this is the most important one. Here looking at the retinas of mice, where we are immunoprecipitating different proteins and PCRing the notch promoter. So we are setting who is sitting on the notch promoter. So in wild-type retinas, you have CBP sitting on the notch promoter. In cyclin D1 knockout retinas, there is very little CBP sitting on the notch promoter. In wild-type retinas, you have acetylation of those histones that are CBP dependent. In cyclin D1 knockout retinas, this acetylation is greatly reduced. So in summary, I showed you a work of Fred Bienveni at the kind of end result mechanistic part. What we have shown is that cyclin D1 is performing transcriptional functions. So to summarize the whole thing, we made a tagged cyclin D1 strain. Then we look at all the interactors. We found that several of them are transcriptional proteins. We mapped the sites where cyclin D1 is sitting. We intersected this with the gene expression profiles. And we found that in the retinas, cyclin D1 is sitting in a key gene, the promoter of the Notch gene, when it turns expression of Notch1 by bringing CBP histone acetyltransferase and promoting acetylation of histones. If we knock out cyclin D1, then there is less CBP recruitment, there is less acetylation of those histones, there is less Notch being expressed, and this seems to be at least partly responsible for the phenotype, because when can we put Notch back into these cells, we can really partially rescue the proliferative defect. And the more kind of global way, I want to say that this kind of approach, when you are inserting flag and HA tags into the gene, now it allows us to take virtually any organ or any cell type from our mouse, and at the proteome-wide or genome-wide scale, we can either build the interactomes, so we can ask what are the protein partners, or we can interrogate the interaction with the genome. Okay, so I think that I talked like 35 minutes. I can stop here and take questions, or we can have a break and we have another 25 minutes lecture. How would you like it? If you're overwhelmed and you think this is enough, then maybe we stop here. If you didn't have enough, we can have another short lecture. I'm fine either way. So in the last part of my fourth lecture, I'm going to talk about cyclin D1, the same protein, but not in development, but in cancer. Because as I mentioned to you, cyclin D1 is overexpressed in over 50% of human breast cancers, and I showed you this slide where cyclin D1 overexpression is seen in the nuclei of cancer cells. And a postdoc in my lab, C1 and Gerard Natai. So today are the stories from guys. Fred Bienveni is a Frenchman and C1 is from Thailand. So he wanted to know what are cyclin D1 partners in cancer cells, not in development, but in cancer cells. And this is C1. He now went back to Bangkok, and he has his own lab in Sirat Hospital in the Mahidol University. So because he wanted to know who are the partners of cyclin D1 in cancer cells, he could not do it genetically the way Fred did, but rather he did kind of a little bit old-fashioned or traditional way, when he takes cyclin D1, attaches to tags, and then expresses it ectopically in different human cancer cell lines. And he took leukemia, breast cancer, squamous cell carcinoma, colon cancer, and breast cancer. And from each of them, he purified cyclin D1 associated complex. This is cyclin D1, and each of those bands correspond to different interactors. And then he repeated rounds of mass spec sequencing. It came up with a socialist star. It looks like a socialist star because there are five different cell lines. And you can see that some of cyclin D1 interactors, like these ones, are command interactors. They are binding cyclin D1 in all five cell lines. These are binding only in 4 cell lines, and there are some specific interactors. For example, this protein is only binding cyclin D1 in colon cancer, and those 3 proteins only in leukemic cells. And then he put all together with our bioinformatics collaborators. This was done by Yido Hu, originally from China or from Taiwan, and he would like to say we put all those proteins and asked to look in each cell in each cell line. So this is leukemia, colon cancer, breast cancer, squamous cell carcinoma. Which functions those interactors, these binding proteins, which functions they denote or which functions they are enriched for. So the green meaning not enriched and red is being enriched. So for example, like this function, of course you cannot see what it is, and neither can I, but that's irrelevant because this is not enriched in any cell line. Here we have red here, so this function, whatever it is only enriched in one cell line, but we are interested in this red top. These are the functions that are enriched among interactors in all cell lines. And those functions are cell cycle, cell cycle, cell cycle, but quite unexpectedly we saw response to DNA damage, stimulus, and DNA repair. So there are some proteins binding to cyclin D1 that play a role in DNA repair. And indeed, when you look at all those interactors and classify them to different functions, you see this kind of little island of the DNA repair proteins. And then there's also a way with our bioinformatics collaborators to make a network when you put all those proteins and then ask to draw the lines between the known interactors, because between the proteins that were shown to interact in the literature. And then this analysis showed us this red island, and this is our interactome network, and this red island are proteins involved in DNA repair. And I'll zoom in here, and right in the center of this network is GAT51. GAT51 is a protein that repairs DNA double-stranded breaks through homologous recombination. So this is all great. We have several DNA repair proteins binding to cyclin D1. So now we want to see whether DNA repair is anyway impacted if we reduce the level of cyclin D1. So now we take human cancer cells, either in the intact form, or we knock down cyclin D1 in red, and now we irradiate them and you look at the survival. So for example, at the foregrays and the control cells, this is the survival, but if you knock down, the survival goes down. So in those HeLa cells, this is cervical carcinoma cells, when you knock down cyclin D1, the survival or the response to the DNA damage becomes more acute, more dramatic. They are responding stronger to the irradiation. Now we are going to use 3 different drugs, and this is the survival. So now we are changing the concentration of this drug, and we're looking at the percent of survival. So if we don't give any drug, then 100% of cells survive. As we increase the concentration of the drugs, the survival goes down. But when you deplete cyclin D1 in red, then the survival is reduced. And of course, we wanted to see whether this is a kinase-dependent function of cyclin D1, of kinase-dependent. So the way to do it is, here we knock down cyclin D1 and then irradiate cells. And as you can see, the survival is reduced. And here, rather than knocking out cyclin D1 or knocking down cyclin D1, we are treating cells with this inhibitor that I introduced in one of the lectures, PD033291, which wipes out the kinase activity of cyclin D1. A treatment with this inhibitor has no effect. So that's kind of interesting, because this is dependent on cyclin D1, but not the kinase activity of cyclin D1 CDK4. So it seems that we have something new, some new function of cyclin D1 that is not dependent of cyclin D1 kinase. But so far, we didn't look at DNA damage or DNA repair. We just looked at survival. We irradiate cells and we look how will they survive. So, of course, we irradiate cells, we damage DNA, but we haven't looked at damaged DNA yet. Now we'll look at damaged DNA, and this is the so-called COMET assay. So in this assay, you irradiate the cells, and then you at different time points, you put them into the electric field, and then you resolve them. And then, if the cell has a broken DNA, it has this tail, and this tail. So at time 0, before irradiation, there is no tail. When you irradiate cells, then there is a tail. This is from the cell. This is the broken DNA leaching from the nucleus. And once it's repaired, then the tail is gone. If this cell has no repaired DNA, the tail is there. So now we are looking at the tail length. And black are control, and red is cyclin D1 knockout. At time 0, there is very little tail. 4 hours, the tail becomes longer, because we damage DNA, but after 16 hours, in black in control cells, the tail became normal, meaning the DNA became repaired, but in red in cyclin D1 knockout, the tail is still big. So there's a persistence of damaged DNA. So if we knock down cyclin D1, then it takes longer time to repair DNA. Now this doesn't tell us mechanistically how it happens, but in mammalian cells there are two major pathways of DNA repair. One is called non-homologous end joining, and this is a non-faithful way of repairing DNA, error-prone, and introduces errors to the DNA. And the second is homologous recombination, which is a very faithful way of repairing. And this homologous recombination, there's a perfect way of repairing double-stranded breaks, is driven by RAD51. And I did mention to you that you actually found RAD51 as a cyclin D1 binding protein. In fact, it was right in the center of those binding proteins. So first of all, we wanted to see, does cyclin D1 really bind RAD51, not through mass spec, but through immunoprecipitation Western blotting. So here we immunoprecipitate cyclin D1 and Western blot for RAD51, not only in the cell lines that we use for our mass spec, but in 1, 2, 3, 4, 5, 6, 7, and 9 different cells, and in each of them we see the interaction. Here we irradiate the cells, and then you look for the interaction between cyclin D1 and RAD51. At time 0, this interaction is very weak. Now, of course, I'm showing you strong interaction because we are exposing plot here for longer. Here we expose for short time, because what I want to show you is that with time, after 6 hours, this interaction becomes stronger. So this is interaction induced by irradiation, and this is at one time point, but at different level of radiation. No irradiation, weak interaction, 5 grays, stronger, 15 strongest. So this is proportional. This interaction is induced by irradiation, and it occurs with time. Now we want to see whether, if we knock down cyclin D1, whether homologous recombination is impaired. And there is actually a very nice essay developed by Maria Jasin. There is a reporter which contains 2 imperfect GFP cassettes which do not express GFP because they is broken by the stop cassette. And there is a restriction site, ISC1, introduced into this cassette. This is the sequence that does not occur in the human genome. And therefore, if you now put into the cells this restriction enzyme IASC1 is going to cut only here, nowhere in the genome, but just right here. And then if this cut is repaired by the homologous recombination, now the cells will become green. So this is the way how you can induce this enzyme, make the cut, and then see whether the cells become green. If they become green, it means that they repair this cut through the homologous recombination. So let's see what happens. If you don't do anything, but you do this cut, then the cells are becoming green. If you knock down RAD51, then the cells are not becoming green, and that's not surprising. RAD51 is a protein that is driving this homologous recombination process. But when you knock down cyclin D1 using one siRNA or the second or the third, then this homologous recombination is impaired. Now, Wojtek Michowski actually asked whether cyclin D1 RAD51 interaction is direct or indirect. What I've showed you is that when you do immunoprecipitation Western blotting, that they bind, but it doesn't mean this is a direct interaction, right? There could be like another protein bridging them together. So Wojtek Michowski took a purified cyclin D1, purified RAD51, mixed them, and found that they bind to each other. So this is purified cyclin D1, purified RAD51, and they bind. And by making different deletions, he actually identified the exact domain of cyclin D1 that binds RAD51, and he can make the mutant that does not bind RAD51 and perform the following experiment. We take the cells, we make this cut, and they are becoming green. Again, if we knock down RAD51, they are not becoming green, because RAD51 is the enzyme that drives homologous recombination. If we knock down cyclin D1 using 1, 2nd, or 3rd siRNA, that is making cells green, meaning homologous recombination goes down, I showed you. Now we do a control experiment. We are going to knock down cyclin D1 and put back wild-type cyclin D1, but cyclin D1 that is resistant to siRNA. And this one can rescue. Well, that's a control experiment, right? So this is all great. But now we are going to knock down cyclin D1 and put cyclin, this is knock down cyclin D1, and then put cyclin D1 that is a mutant. This cyclin D1 cannot bind RAD51, and this mutant cannot rescue. So the wild-type cyclin D1 can rescue, but the mutant cannot, indicating that the cyclin D1 must bind to RAD51 in order to help cells to drive the homologous recombination process. Now, in our institute, there is a lab of David Livingstone, who actually specializes in homologous recombination, and they have a very nice system when they engineered human cancer cells to express this restriction site ISC1. This is the sequence that is not recognized by that is only once in the genome, and these cells also we can put this restriction enzyme and make a cut here. And since we know exactly where the site is located in the genome, we have primers next to it. So we can, by immunoprecipitating your protein and by doing PCR here, you can see, is my protein sitting next to the cut site? You can look before and after. So this way you can look at the recruitment of different proteins just to this specific cut site. And this is done here. You immunoprecipitate RAD51 before and after the cut, and RAD51 should be recruited, should be present after, but not before. This is actually shown here. This is the recruitment of immunoglobulin before and after cutting. Of course, IgG is not recruited. This is a control. This is recruitment of E2F4 before and after cutting. Of course, E2F4 is not recruited to the cut site, and this is the recruitment of RAD51 before and after. So RAD51 is being recruited. A surprise came when Siwalan found that actually cyclin D1 is also recruited to the sites of double-stranded break. So here again, this is recruitment of RAD51 before and after making the break, and this is recruitment of cyclin D1 before and after making the break. Now we wanted to know whether, as we just discussed in the first lecture, whether cyclin D1 and RAD51 are actually co-recruited together, whether they are sitting together. So now we are immunoprecipitating cyclin D1, then re-immunoprecipitating RAD51 and doing PCR, and we see that they form complex. So RAD51 and cyclin D1 are together brought to the DNA after the cutting. This is another way of looking at this. So here we take the cells and we take the laser, like this one, but stronger, and then we make them a stripe, like here. And then this is very dark, but maybe you can see it here. There is phosphohistone H2AX stripe, meaning that this is broken DNA. Oh, perfect, thank you so much. This is broken DNA, and now you can see that cyclin D1 is sitting on this stripe. Can you see it here? This is cyclin D1 on a stripe. So imagine I take this and I, boom, make broken DNA, and then you can see that there's a staining for broken DNA, and there's staining for cyclin D1. So cyclin D1 is so not only cyclin D1 and RAD51 are being recruited, but what happens if you don't have cyclin D1, that RAD51 is not recruited well. So here you can see here, I told you that this is recruitment of RAD51 before and after the break, and see what happened if we knock down cyclin D1. Now RAD51 is not being recruited. This is another experiment. We irradiate the cells and then we look for RAD51 foci. So if we don't irradiate the cells, the RAD51 is not recruited to DNA. Thank you very much. If we irradiate cells, the RAD51 is recruited to DNA. But if we irradiate cells and knock down cyclin D1, the RAD51 recruitment is gone. So cyclin D1 goes to broken DNA and helps to recruit RAD51. To the broken DNA, and this is the model shown here. So cyclin D1 is here, and it helps to recruit. Cyclin D1 is recruited to broken DNA, and it helps to recruit RAD51, and RAD51 in turn drives homologous recombination. If we knock down cyclin D1, then there is less RAD51 being recruited to DNA, and there is less homologous recombination. Of course, the question is, who is bringing cyclin D1 to DNA? Who is this green protein that brings cyclin D1? Well, we went back to this network, and we found that there is a protein called BRCA2 in this interactome. This is a protein that is very early on recruited to the double-stranded breaks. So we found that not only cyclin D1 can bind BRCA2 in the mass spec analysis, but also there's a direct interaction, as shown here by the purified proteins. And now we hypothesize that this is cyclin. It is known that BRCA2 is helping recruiting RAD51. We are proposing this occurs with cyclin D1. BRCA2 recruits cyclin D1, and cyclin D1 recruits RAD51. So I showed you that if we don't have cyclin D1, that there is less RAD51 recruitment. Now we want to see if we don't have the green recruitment, if we don't have BRCA2, would cyclin D1 be recruited? And the answer is no. So this is recruitment of cyclin D1 to DNA before and after the cut. And if we knock down BRCA2, now this recruitment is gone. Also, recruitment of GAT51 is gone as expected. So in summary, this is when you have a DNA break, BRCA2 helps to recruit GAT51, sorry, cyclin D1. Cyclin D1 is recruiting GAT51, and GAT51 is driving the homologous recombination process. So to summarize this part, this was kind of a surprising and kind of counterintuitive, because I told you that the major function of cyclin D1 is the kinase-dependent function of phosphorylating the retinoblastoma protein. And now I'm saying that in the cancer cells, the cyclin D1 performs a CDK-independent function, and this is RB-independent functions. Now, how do we know that retinoblastoma protein is such an important target for cyclin D1? Because in cancer cells, if you knock down cyclin D1, the cancer cell proliferation stops because the retinoblastoma protein is not phosphorylated. If you had cancer cells that lost the retinoblastoma protein, and retinoblastoma protein is tumor suppressor gene that is often lost in cancer cells, then now knockdown of cyclin D1 will have no effect on cell proliferation. And in fact, all the experiments that I showed you today were done in retinoblastoma negative cells. They are done in cells where knockdown of cyclin D1 has no effect on cell proliferation. We had to do it this way, because if you do it in the control cells where retinoblastoma is present, when you knock down cyclin D1, you would block cell cycle progression, and then if you had some effects, as I hope I convinced you we do, on DNA repair, it could be secondary consequence, right? It could not be some function of cyclin D1 on the repair, but rather you're arresting cell cycle, and then because you're arresting cell cycle, you are having some secondary changes. But no, all those experiments were done in retinoblastoma negative cells. For example, HeLa cells shown here, and before and after knockdown of cyclin D1, the percentage of G1S, G2M is exactly the same. This is percentage of G1S, G2M in control cells. This is one siRNA against cyclin D1. This is the second, this is the third. The cyclin D1 is knocked down, but there is no difference in the proliferation of these cells. So this is a function of cyclin D1 that operates in retinoblastoma negative cells. It has nothing to do with the cell cycle function of cyclin D1. So if you take the cells that are retinoblastoma negative, that lost the retinoblastoma protein, as I told you, that now the knockdown or knockout of cyclin D1 will have no effect on those on the proliferation. I showed it to you here. When we take the retinoblastoma negative cell line, we knock down cyclin D1, and they grow the same way. So now I'll show you this in vivo. We take cells, cancer cells, retinoblastoma negative. We inject into the mice under the skin, and then we wait until the cells form tumors. And then we come back and we measure the size of the tumors in different days. And this is in black. Now in parallel, we are going to knock down cyclin D1 in those retinoblastoma negative tumors. And we are going to look at the growth of these cells. This is in red, it's exactly the same. So these cells don't care for proliferation, whether they have cyclin D1 or not, because they don't have retinoblastoma, unless you irradiate them. And look what happens if you irradiate them. If you irradiate, then now you need cyclin D1. These are the control cells, and these are cyclin D1 knockdown. Because cyclin D1 is required for DNA repair. So if you just keep cells without any DNA damage, then you don't need cyclin D1 in the RB knockout background. But once you irradiate the cells, well, now you start seeing a difference. And this is so each circle corresponds to the different tumor. This is without irradiation. When you knock down cyclin D1, there's no difference. But once you irradiate, then now cyclin D1 is required. And this is the summary of this. So in summary, in this part, I showed you a work by Stefan Jerabatnatai, who found that surprisingly in cells, when the DNA is damaged, the cyclin D1 is going to this broken DNA and is helping to repair broken DNA. And this function is a kinase-independent function of cyclin D1, so this inhibitor is not going to help you anything. But we are very excited about this, because the current dogma in the cancer field is is that D-type cyclins, so the kinase activity, are required for proliferation of retinoblastoma positive cells. And the tumors that lost retinoblastoma protein, and again, this is a tumor suppressor gene that is quite frequently lost. For those tumors, targeting cyclin D1 has no point, because these cells are very well established. If you knock down cyclin D1, as I showed you, they proliferate very nicely. However, our work shows that cyclin D1 plays a kinase-independent function also in retinoblastoma negative tumors. But this is a function in DNA repair. And therefore, if we had a way to target cyclin D1 to decrease, unfortunately, it's not going to work through the inhibitor of kinase. We need to have some drug that inhibits the activity of cyclin D1, that here it could have a therapeutic value also in retinoblastoma negative cells, tumors that currently we think are not suitable for cyclin inhibition. It would not affect their cell cycle progression, but it might help to kill those tumors because of the DNA damage. So lastly, this was done by Siwan Njirawat-Notai and Fred Bienveni. Lisa Bex was a graduate student, was a master's student. Agnieszka Zagorzczan was a research technician. This proteomic analysis was done by Steve Gigi lab and by Josh Elias, who was actually a joint postdoc between Gigi and my lab, and now he's an associate professor in Stanford. The DNA repair work was done with Dave Livingstone's lab, and Rick Young lab with MIT helped out with the ChIP-seq. And this is the slightly older version of our lab. And you know, the being overweight is a big problem in the United States, but this is not the case here. It's just this slide is somehow distorted, so we all look like we put up a lot of weight, but actually we don't. This is just this is distortion. And I think maybe I should point out the major players. So where is Fred? Okay, well, I'm sorry. So Fred is not in this picture. Maybe that's why he got work done. But Siwan Njirawat-Notai, this is the person who did the cancer work, and this is Lisa Bex, the student who helped him. Okay, so this was the very last lecture that you heard from me. Thank you very much, and I'm happy to take questions.